Hello everyone and welcome to AP Bio Unit 2 Review Guide. This unit is primarily focused on the cell structure and the functions of the cell. Alongside there is a little bit of cell transportation, but that would be later elaborated on in Unit 4. But the purpose of this video is to basically go through this review guide. I already have the answers written out, but I'm going to elaborate more on them. And if you haven't seen my previous video, it's basically doing the same thing where I try to go in depth in explaining how these biological systems, cells, so on and so forth really work so you can be prepared for your AP test or for your upcoming unit test. So without further ado, let's begin. Number one, what are the differences between prokaryotic and eukaryotic cell? There are pretty stark differences between them, but the ones that you have to know for this class are really the following. Number one, prokaryotic cells are smaller. They're way, way smaller than eukaryotic. Um, there's not really a reason for this specifically. You can't know why. It's just how they've evolved. So just this is something you have to memorize. They're smaller. And eukaryotics are going to be much, much bigger. Alongside this, you're going to see that eukaryotics are going to be in multicellular organisms, okay? So that's kind of like the second difference. They're going to be in multicellular organisms. Humans are only made up, well, are primarily made up of eukaryotic cells. We may have prokaryotic bacteria in us, but that's kind of besides the point. Again, eukaryotic, multicellular. Think animals, plants, fungi, humans. We are all made up of eukaryotic cells. Prokaryotic, on the other hand, is really bacteria and archaea. This is kind of a Unit 1 review, but it's still kind of good to know. The real big hitting question or answer here is the following. Prokaryotics have no membrane-bound organelles. And really this chapter is about organelles and their functions. So, for example, no membrane organelles, I should probably put bound here, and these guys, the eukaryotics, have membrane-bound organelles. Now, what do I mean by that? So think about the nucleus. The nucleus is like this shell, right? Much like how a cell has and I'll draw it out here, a cell has a plasma membrane. All cells do. That's, by the way, a similarity between these two types of cells. But think of a nucleus as a shell, right? Something that is coating it. That is present in eukaryotics, but it is not present in prokaryotics. So most of the DNA, well, all the DNA, is really inside of this shell. It's protected. That's what a membrane does. It protects things. In a prokaryotic cell, you don't have that, and that's pretty significant. That's the most significant difference. In a prokaryotic cell, you do have DNA. Every cell has DNA, but this DNA is going to be free floating around inside typically the middle of the cell, and those are called nucleoids. Um, Now, with that being said, there are extra differences that you could know and maybe might want to memorize. For example, most prokaryotic cells have circular DNA. Prokaryotic cells have circular DNA, whereas eukaryotic have linear DNA. I'm not sure how useful this would be for your current test, but you should know this by Unit 4 and by the very, very end, Unit 6. That is a really big factor between these differences. And what do I really mean by linear DNA, eukaryotic DNA? Let's look at this for example. Eukaryotic DNA, right here, we will see is linear. Chromosomes, they're aligned, right? Whereas prokaryotic DNA, that's not necessarily the case. They actually have a totally different way they function. They're kind of like a circle. They're called plasmids sometimes. So you could remember it as a plasmid. Again, these are somewhat of later content material, but I think you may expect it on this unit. You could. Definitely this content right here. So yeah, eukaryotic DNA is linear, prokaryotic DNA is circular. These are stuff, again, you have to just remember. There's not much I could really say to help you out here. So yeah. Next, I think, how are plants classified? How are animals classified? Bacteria, fungi. I've mentioned this before, and I'll do it one more time. Bacteria are always prokaryotic. They are unicellular. Archaea are unicellular. They're prokaryotic. Fungi, animals, right? Humans, monkeys, whatever. And lastly, plants are all going to be eukaryotic cells. Are they the same eukaryotic cells? No. Plant cells are not human cells. There are differences. But for the purposes of just characterizing these things and making them into two different sections, prokaryotic, unicellular, eukaryotic, multicellular. Humans are multicellular organisms, animals, so on and so forth. Okay. Next, why are cells so small? So if you were to look at a human and you were to compare the human cells to an animal cell that's way larger than a human, let's say an elephant. And this is a question that was a concern for a lot of scientists a long time ago. Now it's kind of common knowledge in the biology community. But if I were to get an elephant cell and I were to compare an elephant cell, and I know I'm kind of going on a tangent here, an elephant cell versus a human cell, would there be a size difference? Maybe, but is that size difference significant? No. Okay? It's not going to be like elephant cells are absolutely huge, which is why they account for their largeness. No. Human cells and animal cells are probably going to be around the same size, actually. The real difference is the number of cells an elephant has. It has way more cells. That means it can, make larger structures, like tissues. However, the reason that cells are small is because of this thing called the surface area to volume ratio. Look, cells have to be extraordinarily efficient. They have to get their jobs done. Cells have to make proteins. Cells have to make sure your body is doing what your body needs to do. And that requires sometimes eating food, breaking down material, building material, and all that stuff has to get into the cell and out of the cell. So, scientists have found as you increase the size of a cell, not only does the volume increase, because it's taking up more space, but so does the surface area. Now, as a result of that, would you expect the cell to be more effective or less effective? It's going to be less effective. The reason for that, okay, is that remember, cells have to take in things and take out things. The larger you get, typically the more demand you have, the more things you have to do. So the more stuff has to go in you and the more stuff has to go out of you, right? Larger cell, think of more functions. The problem with that is though, is that as the volume of a cell increases, so as the volume of a cell increases, the surface area increases too, but not at the same rate. The surface area actually increases at a smaller rate than the volume increase. And the volume increases a whole bunch, but the surface area doesn't increase as much. So you have a very odd surface area to volume ratio, where the volume is going to be way more than the surface area. And that is a problem. You don't want cells like this, and that's why most cells are not large, because the volume is so huge, the demand is so high, but the surface area is kind of small in comparison to the volume, and therefore not a lot of things can actually get through. Or if they do get through, it takes a way longer time. Imagine if I want to take a protein and I want to bring it all the way to the nucleus that's located in the middle of the cell. That's going to take a load more time to get all the way here, whereas if I have, let's say, this smaller cell, right, that's right here, it's going to take way less time. So I hope that kind of makes sense, that you want a cell that is small, but you want a cell that has very high surface area, because you want the cell to be able to take in as much as it can and release as much as it can, so it can ramp up its efficiency. That's how your body really gets things pumping. That's how you typically see smaller cells in a human body. That's why elephant cells are not much larger than a human cell either, because they want you. We are naturally programmed to have a cell that has a high surface area to volume ratio. The surface area is like 18 to 1 sometimes. It's an exaggeration, but it's something you should remember. So, as I've recorded here, you want a higher surface area to volume ratio. Okay? That is the most important part, and I will actually highlight it. Okay? And that's also why cells are small, because their surface area to volume ratio is super high. How is the folding of the inner membrane, which increases surface area, beneficial to the cell? So this is typically going to be seen in the mitochondria. If I were to Google the mitochondria structure right here, you're going to notice two things, right? You're going to notice this green layer right here, and ignore the red stuff, but you see these like folds? These folds are called cristae. This is going to be something important in Unit 3, but again, it's good to become familiar with it. I'll go to like another photo right here, but it's all these folds. And if you think about it, as you make folds, you can fit more things in a smaller area that increases your surface area. And remember that on the membranes of all these cells, right? Of all these cells, right? On their membranes are going to be proteins. So on this cristae, there's going to be proteins. So if I am able to, in a way, fold everything and make them small, 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 like this, right? I'm going to be able to fit a lot of proteins on the surface area, or on the surface, or on the membrane of my cell. And if I'm able to put a lot of proteins on the membrane of my cell, I can do more things efficiently, right? Because I have more proteins. More proteins means they're going to be functioning better. And also, there are more opportunities, more areas for other things, like let's say a hormone, to kind of reach my cell. There's more contact areas. So that just overall adds to the efficiency of the cell. It makes sense why the mitochondria has a fold, because this is the powerhouse of the cell, right? It makes energy for us. So we want to ramp up our energy production. We really want to be as efficient as possible. So think of the mitochondria as, a great example of folding. It's called the cristae. It's the second layer of the mitochondria, but it explains why um, we have it. All right, it's just a total use of efficiency. Okay, and I guess an overarching theme, I guess, in all parts of cell structure is efficiency, efficiency, efficiency. You always want efficiency. Okay? Cool. So, where is it? Yeah, right here. I have already highlighted this. The cell membrane is highly flexible, and the components are able to flow around the surface of this cell. What do I mean by that? There is the cell mosaic, or the cell membrane mosaic theory, which I won't really get into because it's a long story, but I will try to show you what the cell membrane looks like. Okay, cell membrane diagram. Okay, now if you were to look at this, these all seem fixated in place, but that's not the reality of the actual cell. Molecules move. Okay, all molecules are moving around, and your cell is nothing but molecules. Now, are these molecules kind of in place? Yes, they are, but they are subject to moving around. If I were to get, let's say, one molecule here and one molecule here, you should expect them to be moving around in a certain degree. They're not just fixated in place. They're somewhat flexible. Okay, so that's something you kind of have to remember. But it's just the way molecules move. And if I heat the molecules up, they'll move faster. If I uh, if I were to cool them down, they move slower. Okay, so next, number 5. The structure of the cell membrane results in selective permeability, which allows cells to have an internal environment that is different from its external environment. I think right now, I think it would be a really good opportunity to kind of just dive into the plasma membrane of the cell, because it is one of the most important parts of this chapter. So, the plasma membrane of the cell is really just composed of two things. It is composed of proteins, and most importantly, phospholipids. And most of the cell's um, typical features are going to be based off the phospholipids. And there's different types of phospholipids that you'll see that will impact the way the structure or the properties of the plasma membrane are. So let's pull up a phospholipid diagram here, and let's just really get to analyzing it. Um, and I will take a screenshot of this right here, boom, and really try to get into the nitty-gritty. So a phospholipid is basically a lipid, but it has this group right here called the phosphate group. It is a functional group that increases reactivity. And if you were to think about it, this has a negative O right here, and that is significant. It has an R group, which, if you remember from proteins, could be polar, could be nonpolar, so on and so forth. But it's really this O right here and this phosphorus bond right here that make this a very significant thing. It makes it hydrophilic, right? Because it's charged, and it's going to be trying to bond with with positively charged regions of another molecule. That's why this is significant. So the heads of the phospholipid are going to be actually pretty hydrophilic as a result of this. Hydrophilic. Okay? Now, if you were to look at a phospholipid bilayer, the way the plasma membrane is structured, you will notice that all the heads are on the top of the structure and all the other heads are at the bottom. Why is that? Why are all the heads of the phospholipids facing upwards towards the exterior of the cell or to the interior of the cell? Well, for one reason and one reason only, because they also want to protect the cell in a way. Um, you don't want everything that is in your body to be going into different cells, right? You want to protect the cell in general. So this is a form of protection, okay? It creates selectivity, okay? The hydrophilic regions keep the most of the water outside of that hydrophobic region. And the hydrophilic regions at the bottom keep the inside of the cell kind of in place because it prevents them, well, the hydrophobic interior right here prevents them from going out, okay, unless regulated. All right. Now, if you were to look at this, it makes sense though. All the hydrophilic portions are shooting up towards the exterior of the cell where water is typically located. So that's great. That makes sense. And all of the hydrophilic tails at the bottom are towards the inside of the cell because the cell has cytosol, which is a polar molecule. Okay? So because of that reason, it explains why we have a bilayer, why the tails are in the middle of the region of the bilayer and why the heads are at the top of the bilayer. Okay, that is a very key characteristic of the actual thing. Next up, what you're going to be seeing is how these things kind of interact with one another. If you kind of remember a little bit to chemistry, are the phospholipid bilayers 100% bonded to one another? No, they are not. What they are actually forming are these things called weak interactions, where the O negative here is probably connected to a positive, like let's say we imagine another chemical right here, another phospholipid, if I were to just draw it, right? This is typically how they draw it in AP Bio 2, by the way. And it's going to have like a little bit of a positive region here, maybe a negative region here. This positive region is going to slowly bond. It's called a dipole-dipole bond. You don't really have to get into the chemistry of it, but just know that the hydrophilic regions are attracted to the hydrophilic regions because part of them is positively charged and part of them is negatively charged. On the other hand, you have these hydrocarbon tails right here, and those are extraordinarily significant to the way that these phospholipids actually bond as well. See, the hydrophobic tails, although they don't have anything that can engage in polarity, have this thing called a van der Waal interaction. A van der Waal interaction. Now, I'm not going to get too much into the chemistry of it, but there are moments in a nonpolar bond. Assume that this is oxygen and oxygen bonded together. It is a nonpolar bond. There are moments in this bond where electrons, right, the things that make the atom negatively charged, gravitate a little bit towards the one atom, because electrons are free moving, by the way. They are free moving. They're not necessarily bound to the atom indefinitely. They can sometimes shoot out, they'll come back. It's weird. Don't get too much into the chemistry, but if I have a bond that is nonpolar, that just means that these guys have relatively the same charge. But there are small instances, momentaneous instances, where electrons that are being shared typically end up a little bit closer to one element than the other one. And when that happens, one of the elements becomes slightly more negatively charged and the other one becomes slightly more positively charged. And because you have this slight difference in charges, you are going to end up creating some form of polarity, and then these hydrophobic tails are going to be able to bond with one another. And that kind of makes sense if you look at, this diagram right here. How are these two tails right at the bottom really just kind of connected? It's because of these van der Waal interactions. These are very momentaneous, by the way. They happen and then they go away. They happen and they go away, which also explains, by the way, why the cell membrane is a fluid moving thing, because these bonds are constantly and they're not bonds, they're weak attractions from charges. They're electrostatic charges, positive and negative charges, that occur very momentaneously. They break, so the molecules actually have a little bit of free movement. Now, because they're always in constant contact and there's a lot of phospholipids, they kind of stay bumped together. But for the most part, yes, they are somewhat free moving. Okay? So that is the general structure of the phospholipid bilayer, but there is a little bit more to it than just understanding the idea that there is a hydrophobic interior and a hydrophilic exterior. Okay? So what else is in this? There are proteins and cholesterol. Okay? Those are the other two things that are, and I'll draw a protein like this, and cholesterol like this in the cell. Okay? Now I'm going to explain the structures of these proteins and the structure of the cholesterol, and then I'm going to go more into the property and the selectivity of the membrane. So like I said, in the plasma membrane, and I want to actually extend this protein, so ignore this right here. Just assume that this is one large protein. Um, so remember that in the plasma membrane, these hydrophobic tails are slightly bonded with one another. These hydrophilic ends, the tips, are very, very much bonded together because of polar attractions. But now you have to consider the other things that are in the plasma membrane, which is cholesterol. Cholesterol. It's also a lipid, and a protein, which is not a lipid, is its own macromolecule, if you remember from Unit 1. So, a protein, in order for it to be on the plasma membrane, must have two characteristics. It must have a hydrophobic region and a hydrophilic region. And it kind of follows the same structure as these phospholipids. The hydrophobic region of a protein on the plasma membrane is typically going to be around the interior of the protein, on the rim of it that's connected towards the tails. Right here, all of this is going to be probably hydrophobic. Hydrophobic. And it makes sense because it wants to kind of ferment and cement itself in there. Proteins don't want to move around. They don't want to leave the cell if it's supposed to be on the membrane. They want to be stuck there. That's how they're going to be structured. They're going to be having a hydrophobic region on the exterior, and also at the very um, top or the bottom of the exterior, they're going to have this hydrophilic region, and that just helps stabilize their bonding. Okay? Now, when you look at the interior of a protein, so if you were to imagine a protein kind of like being like a little tunnel, the interior of the protein is typically going to be hydrophilic 98% of the time. And I'm going to explain why momentarily. But just understand that for proteins, their exterior is going to have a middle region that is hydrophobic, and a bottom and top that are pretty hydrophilic. And then you're going to have an interior that is almost always going to be an interior, which is another section of the protein, an interior which is always going to be hydrophilic. And I'll explain why momentarily. Okay. And if you were to see this, look at these proteins. These rims right here, right, where you have these like phospholipids, this is going to be hydrophobic. These very edge rims are all going to be hydrophilic, and this interior, this darker shade blue region right here, they're all going to be hydrophilic. Okay? So with that being said, let's move on to cholesterol, and I will just pull up the diagram of cholesterol. I can't spell it. I'm illiterate. My apologies. Cholesterol looks kind of crazy, but it's not too bad. I want all of you to basically recognize this one important part of cholesterol. This one big important part. That cholesterol is chunky. This is a very thick molecule compared to a phospholipid, which is relatively thin, right? It just has a tail. This is like made of rings and rings and rings with, it's just complicated in a way. And it's really composed of two portions, kind of like a phospholipid, composed of a hydrophilic portion, this OH, which can hydrogen bond, apologies for my poor handwriting, and this basically super duper hydrophobic region as well. So if you were to then pull up in a cell membrane, you would notice that the cholesterol looks kind of like this. Okay, this is cholesterol. And cholesterol, if you see these membranes or these rings are always going to be in the interior, but the top part of it is going to be sticking out, and that top part is this OH group. Okay, so these are the components of a cell, and these cholesterols are going to be pretty resistant to movement. Okay, now I'm going to be talking about the significance of, or kind of, now that I've explained what is in the cell, and this is, by the way, is the best example. This is a really good picture by Khan Academy, actually, the mosaic model of the cell. This is what it looks like from what I tried to draw. But now I'm going to be talking about the aspects of the cell, right? What can change its mobility or its fluidity? Remember, I said that the molecules are kind of moving around, but you can actually break a plasma membrane, or you could really solidify it depending on what you have inside of it. Okay? And I'm going to get into that right now. So, how to increase fluidity or fluidity of a membrane. 1. Temperature. Temperature. Always going to be temperature. Super key right here. So temperature, what it makes, or what it does, is it increases the speed of the molecules. So if you were to basically imagine two molecules right here kind of attracted to one another, right? And let's just say that this is the heads that are hydrophilic and the tails that are hydrophobic. They're going to be attracted to one another, so they're going to be pulling themselves a little bit towards one another. These are the forces of the molecules combined together. As I increase temperature, though, the molecules are going to be moving around a lot more. Sure, they will be bumping into each other, but the biggest point, though, is that because they're hitting each other so quickly, these attractive forces actually get nullified. Because imagine if you have your two hands together and you basically hit them together so quickly, they kind of recoil, they bounce off of each other a little bit. That's kind of what's happening when you have two things so quick hitting one another, right? That they end up actually bouncing off. That destroys fluidity. Okay? This is when you increase temperature, by the way. You're destroying fluidity, destroying it when you increase fluidity. Or when you increase temperature, my apologies. If I were to reduce temperature, basically really prevent this plasma membrane from moving around, or from the molecules moving around, I'm actually going to get a very concrete membrane where things can't really go in and out. And the reason for that is that as I freeze everything, okay, I will essentially be putting them in place. And their forces, those bonds, those attractive forces that I was talking about, will really be able to solidify, or really manifest themselves and really just stick these things together and just make it like glued together. That is going to be worrisome because you're not destroying the membrane, but you're preventing things from that would naturally get in the membrane without a protein to not be able to get it. So for example, nonpolar molecules, and I'll explain later, don't worry, nonpolar molecules will kind of easily go through, that are very small nonpolar molecules can easily go through a membrane without any protein or anything needed. But if I have a very low temperature, when everything is kind of bunched together, I'm actually going to be unable to get that molecule through, and that's going to hinder the cell. Okay, so that's number one. Number two is unsaturated fats, unsaturated lipids, really. So if you recall, the phospholipid has what? The phosphate head and that lipid hydrocarbon tail. So if you recall from Unit 1, there are saturated and unsaturated fats. Basic review of them is that a saturated fat, basically, and I'll pull up unsaturated fat. An unsaturated, chemical structure, my bad. Perfect example. If you look at the saturated thing, all the hydrogens are connected to it. On the other hand, an unsaturated fat does not have these two hydrogens attached to it, and it has this double bonded carbon. that's problematic because it creates a kink. It actually extends or it moves around the molecules. Okay, a little bit. It gives it more fluidity because it's not like kind of rigid. And the problem with that is if you can imagine two molecules like imagine these are the lipids, right? The hydrophobic lipids, the hydrocarbon tails, hydrophobic, right? Imagine these are saturated. And by the way, it varies. The saturation varies in a cell. These are hydrophobic lipids, and I have one that is, and imagine this is a unsaturated fat, and I have these two that are saturated, right? The fact that I have a kink in it may sometimes be beneficial because it could decrease the distance between the two molecules, or sometimes it can work against your favor, and it could work the opposite. It could be like that, and the distance will change. Why is distance important? Because if I keep my hydrocarbon tails away from each other, right? If these guys can never touch each other, their van der Waals interactions won't happen. Think of it, if you want to make a bond, you have to make two molecules kind of combine or collide. Well, if these tails are not going to combine or collide, then they're not going to be able to basically fortify their forces together, right? So it's going to be super hard to do that, and as a result, if you can't get your molecules to be close together at their hydrophobic tails, they're going to be much more fluid. They're going to be fluid because the forces can't stabilize, right? They can't kind of come together and allow that nice structure, that bilayer to form. Things are going to kind of move around more freely. So number two really is unsaturated fats, unsaturated tails. Okay, that's a big thing. Next up is going to be tail length. Tail length. Now, I'm not too sure if this is going to be as important, so you could probably skip this part. But for people that are, on the edge, maybe want to know, this part is just kind of intuitive. If I have a very small tail, right, between these two things, and just assume that they're saturated fats, if I have a very short tail, I can only make fewer bonds, right? I can only make, let's say, 3 bonds, whereas if I had a long tail, and I'll use it on the left side, if I had long tails, I can make, maybe 4 bonds, 5 bonds. And the more bonds you have, by the way, when I say bond, I mean weak attractions, right? These are those electrostatic van der Waals attractions. The more length I have, the more weak attractions I can make, and the more stability I can get. So tail length can vary that. Weaker, or smaller tails, smaller tails equal weaker or more fluid membrane. And a larger tail equal a more structured or rigid. I'll call it rigid membrane. Okay. So that's that. And lastly, and this is an important part, is cholesterol. Number 4 is cholesterol. Okay, cholesterol impacts the mobility of everything again. Why? Okay, so if I were to draw my hydrocarbon tails, right, and my hydrophilic heads, and I were to draw that OH right here, and then that really big chunky model of cholesterol. Let's say I were to increase temperature. All right. Now remember, when I increase my temperature, I make my thing much more fluid. But because this is a very chunky, large molecule, right? It is going to be more resistant to temperature change because it has larger forces. Bigger molecules typically have larger forces they exert. Think of it kind of like gravity, even though it's not like gravity. The more mass you have, the more pull you have. It's not exactly how it works, but you're in biology, you don't really need to know that type of stuff. That's more chemistry. So this is going to be less resistant or more resistant to temperature change. So as these molecules are moving around, bouncing around each other, the cholesterol, in a way, actually keeps the mobility in check. It's actually helpful in this way. If temperature increases, it actually reduces the amount of particle movement because it keeps the molecules kind of confined in a space. Okay? It doesn't allow the molecules to kind of go bounce around and do whatever the hell they want. It really just ensures that these molecules stay where they're supposed to be. And in a way, then, cholesterol, right, in a more rigid in a more fluid environment or a hot environment, serve to decrease, and I will say hot, right? A hot environment serve to actually decrease, okay, mobility. They keep things in place. They keep things in place. Vice versa. When I decrease my temperature, the way the things interact actually prompt the molecules to kind of be moving around more. So if I have cold temp, and by the way, you don't really have to know stuff for cold temperature, just know it for high temperature, the impact of the cholesterol. It kind of acts like antifreeze. That's a great example. I don't know how antifreeze works, but I remember that was in the textbook. So if you know how antifreeze works and you can kind of connect it to that, you're welcome. If not, well, congrats, you're like 95% of the population in trying to compare these two things. So, yeah, cold temperatures, it actually reduces rigidity. So just kind of think of these things as working against one another. Um, Again, this is something you kind of have to memorize. Unfortunately, I don't really. It's not properly explained. So now that we understand that, and I guess I should do you the favor and write everything that impacts mobility of the plasma membrane. One, temperature. Okay. Cholesterol is the second most important thing. Then you have the unsaturated fats, and then finally fat or lipid tails. And then lastly, you're gonna have the tail length, right? And this is probably in order of importance. You're gonna see it on the AP test. So now that I've explained this portion to you of basically how is this entire plasma membrane structured, I'm going to kind of explain what molecules can go in and what molecules cannot go in. And that's really going to be most of the talk about the plasma membrane, and after that we're going to probably go into all the structures and transportation systems of the cell. Okay? So, the plasma membrane, if we were to get a photo of it, right here, boom, get it for that. If we were to get a photo of the plasma membrane and look at it, we're going to have to think to ourselves right now, okay, what can get in? Well, small molecule, and I'm going to make a ranking system right here. What gets in naturally? This means that it does not require anything else to get into the cell. What gets in naturally into the cell? Okay? Well, the most easiest thing that gets in are non-polar small molecules. You must be asking yourself, well, how is that? Why does that work? Well, the hydrophobic region of the plasma membrane actually constitutes the majority of it. I mean, simply look at these photos. A lot of hydrophobic region and a very small, well, in relative term, a smaller amount of the hydrophilic region. In addition to this, these scales are exaggerated. They're very, very much exaggerated. These tips, or these heads right here, are not circular really. They're somewhat they are, but in reality, they're very, very small. There's actually gaps in between all of these things. So very small molecules can actually fit through these gaps and really be able to get through. It's going to be super, super easy for them. Okay? Next up, you're going to have non-polar molecules that are larger, non-polar that are large. They'll get in, but it's going to be taking a little bit more requirement because now they're going to be interacting a lot more with this hydrophobic or hydrophilic region. And remember, hydrophilic does not like hydrophobic. They do not like one another. Okay, so when they kind of hit each other, they're not going to really interact. They're going to kind of push one another away. So will the larger things get in? Yes, because the hydrophilic interior is still the majority of the plasma membrane, so it's still going to be favorable. It's kind of just going to push its way down. It's going to be cool. It's going to be dandy. So that's the second thing that'll kind of get in with some ease, or, well, a decent amount of ease. Non-polar small molecules, super easy. Super easy. An example of this is going to be O2 or CO. Stuff like this, I don't really know large non-polar molecules, so Best of luck to you guys. You can just Google that right now while you're watching the video. Next up are small polar molecules. These stuff could get in, but it's going to be super hard. Yes, they can probably pass through the little creaks that are present between those hydrophilic heads, right? Again, remember, this is not the best photo, right? Because the heads are not actually fully touching each other. There are regions where they're kind of like open. They'll get through these heads. They're going to not really like this hydrophobic interior, but because they're small, they could probably fit through it. It's going to be hard, though. It's going to be what I would say it's pretty hard. Okay? Then you're going to have large polar things. And these guys are going to have a really, really hard time. Doesn't mean that they won't get in. No, they could get in, but it's going to take a long time to get all of the large polar molecules through. What is a large polar molecule? Sugar. Okay, what is a small polar molecule? H2O. You can have, I think, urea, ammonia, all these things. And lastly, the hardest thing to get in are ions. Why? And if I were to think of an ion, I think of K+, NaCl, or H+. These things are super duper hard to get through because they're literally charged. They are the definition of a charged molecule. Yes, they're small, right? They can actually probably fit through the little crevices between these two things, between these two hydrophilic heads, but they are so polar that when they go through that hydrophobic region, they're just going to bounce off one another. They're going to go crazy. So they are not going to go in, okay? But the cell has a solution to all of this. The cell says, okay, how do I get the stuff that I really need, like water, right? If water is going to have a hard time naturally going through the cell, how am I going to get it in here? It has those proteins that I was talking about, all right? And I told you I was going to get back to the reason that the interior of the proteins are going to be hydrophilic, right? And the top part are going to be hydrophilic too. These proteins, for water, it's going to be called an aquaporin. Remember that water has aquaporins. There are going to be proteins that actually allow for these polar molecules to get through, and that is why their interior is polar, because it can actually allow them to attract to one another and go down that stream. Okay? Now, there's different types of proteins, but these are in general called transport proteins. I'll get on about transport proteins later on in this video, but you have your first introduction to transport proteins, okay? But in general, this is the ranking of things that can and can't get through the plasma membrane. That word of getting through is called diffusion, by the way, and I'm going to start using that throughout the video. So, you have all that. Next up is what are the structures of all of these things, and what do they do in the cell? Plasma membrane. What does a plasma membrane do? It really adds for protection, right? I want to make sure that I want things that are supposed to go in my cell to actually go in. That's why we have this thing called selectivity. That plasma membrane, if you think about it, is selective. It favors nonpolar molecules that are small. That is selectivity. It wants nonpolar molecules to go in. It doesn't want large polar molecules to go in because typically large polar molecules are proteins. And let's say you have a virus coming into your body and attacking you. It's probably going to be made of RNA, or proteins, and remember, RNA is polar, proteins are polar, so it actually kind of wards itself away from that. That's awesome. That's great, okay? So the plasma membrane is selective to protect the cell, okay? That is the biggest purpose of the plasma membrane, okay? Next up, you have the cytosol or the cytoplasm. Super easy. Basically, it's a jelly-like substance. It's a liquid that basically fills up the entire cell. If I were to show you a photo of a cell, right? Okay. All of this is suspended in a liquid. So if you were to think about like a pool, and I were to throw like balls into the pool, you can think of the balls as the organelles, the organs of the cell, and that water, the pool water, just simply being the cytosol, the cytoplasm. Okay? The nucleus. The nucleus contains the DNA. It acts as like another form of a membrane. It is a membrane, actually. It is formed as a membrane, but it has it's a structure that really just ensures only specific things can go in and out of the cell. Remember, this is where your DNA is. It's gotta have really high selectivity, and it does, by the way. Your nucleus of your cell, I'll prove it to you right here. has these things called nuclear pores. These little circles. Well, that's a lysosome. Where is it? So this is the nucleus. This really dark purple thing right here. It's called the nuclear envelope. It has these very small things illustrated here called a nuclear pore that basically allows only certain proteins to get through. You only want certain proteins to get through. Next, in your nucleus, you have this thing called the nucleolus, which is the most active part of your nucleus. This is where let's say, for example, your kidney cells are actually making the kidney proteins. Remember, your DNA has a lot of information, and it is not making every single protein at that given moment. It is only making certain proteins that are required for that given moment in time. So the nucleolus is that region. It typically is characterized with making a ribosome, and I'll go into literally what a ribosome is in just a moment. But yeah, nuclear pore, nucleus, super, super selective. We want to make sure our DNA is not damaged, right? Because a lot of stuff can go wrong if our DNA is damaged. Think of cancer. Smooth ER, rough ER. Okay, so the smooth ER and rough ER are basically attached to the nucleus. If I were to show you a photo of a cell right here, a human cell, make it easier. Perfect. This green structure right here is basically your smooth and rough ER. What is the biggest difference and what do these functions do? Rough ER is called rough ER because it has ribosomes on it. So think of rough, right? R as ribosome. Okay? So it has these small little ribosomes attached to it. Okay, what do ribosomes do? Ribosomes are protein factory machines. That's all you really need to know about what they do for this current moment. But the significance of a rough ER is the following. It creates proteins that are intended for excretion of the cell, for proteins to leave the cell. What could this be? A hormone protein, right? A signaling molecule, okay? And remember this, a signaling molecule, right? So proteins made by rough ER. The proteins made by the rough ER. Proteins made by rough ER. Anything meant to leave the cell. So an example of that would be, they're called excretory proteins. Excretory proteins. Excretory. And think of them as signaling, antigens. These are just some stuff that you should kind of have a little bit of memorization for. It's not super important, but it's just, you have to know that rough ER 99% of the time makes excretory proteins, and there are things called rough ER ribosomes do that, okay? Then there are things called free ribosomes. These ribosomes are not connected to anything. They are just suspended in the cytoplasm. They make proteins that are for the cell, that are intended for the cell. Okay? Intended for cell usage. And the best example of this would be DNA polymerase. Your teacher would be proud of you if you said that, because you're not even exposed to DNA polymerase. You could say mRNA polymerase. But just in general, intracellular usage is by free ribosomes. They are suspended into bound ribosomes are the terms for the stuff that are made in the um, that are on the ER. Okay. So with that being said, what is a smooth ER? I know I kind of just sidetracked a little bit. Smooth ER is also attached onto the nucleus, but it's not for protein usage. It's actually for lipid creation. Lipid creation. Detoxification of certain molecules. Detoxification. It adds OH. Actually, no, that is the wrong thing. No. It's just there for detoxification. So if you drink alcohol, it's going to go through your smooth ER. And typically, your liver loves or has a lot of smooth ER. So you can actually identify, and here's a very key point I want to make here. You're actually able to identify the function of a cell depending on the concentration of certain things. So if I were to have a cell that has a lot of smooth ER, I know that it's really for detoxification because smooth ER detoxifies things, or I know it's really made for lipid creation. On the other hand, if I have a cell, and this is by the way a test question probably, if I have a cell that has a lot of protein excretion, I know that its bound ribosomes, those things on the rough ER, are going crazy. Okay, so know that. That's just identification. Or if I have a cell that has, I don't know, a lot of stuff breaking down, like protein or digestion, right? I'm gonna probably know it's gonna be a digestive cell, right? Something in my bladder. But we'll go on later into that. So yeah, that's really the difference between the smooth ER, the rough ER. Um, Next up, ribosomes. I kind of described what they are, but there's one part that I didn't. Ribosomes are made up of a thing called rRNA. It is a specific type of RNA and also proteins. All cells have ribosomes. That is actually what links us, what links a plant cell to a human cell, vice versa. Okay. Next up, your mitochondria. Mitochondria, for the purpose of this unit, makes ATP. That's all you got to know. But there's a special thing about mitochondria. Mitochondria has two membranes, okay? One here, and one is the cristae. Boom, 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 boom. All right? Two membranes. Remember that. Super special, makes ATP. That's the degree that you need to know for this class, this unit right now. You'll get much more in depth about this next unit. It's crazy that you'll go into depth for this. So yeah, remember the interior right here is called cristae. That's a good thing you need to know. And it's folded. Remember that folding increases surface area to volume ratio. Stuff that I just talked about earlier in this video. So yeah, that's all you gotta know for right now. Golgi. Golgi is super important. Okay? Golgi is typically got two regions of it. So it's like this weird structure that's kind of made like a layers of layers of layers. Okay? Now the Golgi has this thing called the cis face and the trans face. And it's just the orientation of the Golgi. It's not like super important that you have to know it, but just know that when you have your cell, remember that the rough ER and the ER in general is attached to the nucleus, okay? And you have your bound ribosomes here chugging away making proteins, right? Boom, 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 boom. And as you make these proteins, you kind of have to tell these cells where the hell to go, right? Or not these cells, these proteins, where the hell to go, right? I mean, if I make a protein and it just has no direction, then there's no point of telling it what to do. So what happens is the following. The cells will be launched through this, the rough ER has this membrane, okay? And you should kind of know this. It has a membrane, so does all the organelles in a eukaryotic cell. By the way, remember, the stuff I'm talking about right now is only in eukaryotic cells. They're not in prokaryotic cells. Except not the nucleus, sorry, the ribosome. But it'll launch out of the rough ER from its membrane, and once it goes from there, it's gonna be going into the um, Golgi complex. The cis face is the side that faces the rough ER. Simple as that. And as the protein goes through each layer of the Golgi, it's like the packing center, the packaging, like the UPS, like where is this protein going to go? So as this protein goes through the Golgi, what ends up happening is the following. Each layer has its own unique protein attached to it, right? So imagine like you go through like it's like a factory, right? You have a box and it goes through, station A, station B, station C. And in each station it gets like, go here, go here, go here, or it adds onto it like, let's make the box a little bit bigger and blah 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 blah. Typically once you've reached this end portion of it, okay? What happens is the protein will end up being in here. Let's just think of this 8 as the protein, and as it's going to be leaving the Golgi, right? it's going to be going into a thing called a vesicle. Okay, a vesicle is just a bubble of fat, basically, that is a protective layer for the protein. And what happens is that that vesicle also gets marked. Basically, hey, go here. Okay, and this signal is probably another protein being attached to it. But as this signaling molecule gets attached to it, it'll be recognized by the receptor of where it's intended to go, right? So let's say the receptor is in another cell, right? That's going to have a receptor molecule that'll recognize that little, the vesicle it's called a vesicle that the protein is in, and then and the protein it's literally the vesicle is a phospholipid bilayer. It's super small, okay, but it's just like a little bubble, right? Think of like a hamster in a bubble. That's kind of what this is. It'll go outside the cell eventually and reach this signaling molecule and then shove the protein into the cell. And I'd like to also note that, and I forgot to, my apologies. Once the protein has been made from the rough ER here, and it manages to leave it and go into the Golgi, leaving the rough ER, it actually ends up going into a vesicle as well. It's part of the it's part of the rough ER's membrane. Remember, I mentioned that the rough ER has a membrane. The membrane, all membranes in the cells are always made up of phospholipids, except the one in the nucleus. Okay, it's like a little caveat that I forgot to add. As it leaves the membrane, it's going to go into a vesicle from the membrane of the rough ER, and then it'll be transported into the Golgi. And as it's transported, as you have that protein and it's in its vesicle, the vesicle will be absorbed by the Golgi, okay? And the protein will then just go through its natural pathway down the Golgi, and shoot out its transphase, go into another vesicle. That vesicle gets marked as a with a signaling molecule, and that's ready for exportation of the molecule. Um, so yeah. Next up, lysosome. The lysosome is a suicide sac. It also is for digestion. 95% of the time, it's for digestion. But um, the lysosome is this like small region of your cell. Like think of like one of these like super duper small molecules, and basically it's attached to your vacuole. A vacuole is your food molecule, or your food, your nutrient center. Okay, so you have a cell here, and I have these vacuoles here that contain food, and I know I'm jumping ahead a little bit, but it's kind of necessary to understand the function of a lysosome. And the lysosome is going to basically attach itself to the vacuole. This region will kind of open up a little bit, and the food will kind of go into the lysosome, and the lysosome itself has these digestive enzymes that just break down the food for you. Now there are issues with this, right? Not breaking down the food, but the lysosome in and of itself. And if you were to kind of ignore this, the lysosome can sometimes blow up. And when it blows up, it can literally release a ton of digestive enzymes into your cell, and those digestive enzymes don't discriminate against anything. They'll just go digest whatever the hell they can. And that's a problem because that's what causes your cell to commit suicide, in an indirect way. There's another way called apoptosis. You'll learn about that in Unit 4. But in this way, the suicide sac is just going to start digesting other things, num 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 num. It's going to eat it away. And eventually, if a large amount of these digestive enzymes are released, your cell will die. If a small amount are released, fret not, your cell has repair systems that will come in handy. But a large amount gets released, your cell will die. Okay? Okay. Cell wall. Cell wall is only in plant cells. It is made of cellulose and it allows for protection. It's kind of this sticky like structure. It's basically, if I were to look at a plant cell, it's used for protection, by the way, similar to how a plasma membrane is. But it just sticks onto the plasma membrane. It's nothing super crazy. Um, Pretty strong, gives good protection. And also, it allows for things like water to adhere to it and kind of move up the cell, through capillary action, but that's not as important for this unit. Chloroplast, it is, again, going to be really described in Unit 3. It's like one of the most central things of your unit, but, yeah. The chloroplast is made up of kind of two membranes. It is one of those things, again, where, like I said, all the organelles in the eukaryotic cell have membranes, but this one has two, whereas the other ones only have one. It is made up of two things. One, it's regular first membrane, and then the second one made up of vacuoles. You're going to be more concerned about that in chapter 3, but these vacuoles are like literal stacks of of like membranes. So this is what a chloroplast looks like. It's got these like big stacks of circles, and it's got its like overall outer membrane. It's composed of stroma, which is this liquidy thing. It's basically just like the cytosol, but it's just a little bit different content. That's all you should really remember for this unit for chloroplast. Not that crazy important, except for one specific part. Then you have cytoskeleton. Cytoskeleton provides structure. Really, like literally, it's your bones. It provides structure. It's the skeleton. Well, if you think of a car, it has a cytoskeleton, right? It has like this, the framing of it, right? Protection for um for the molecule. And really interestingly, there are three types of cytoskeleton. There's microtubules, microfilaments, and there's one more. I think it's called intermediate filaments. And your microtubules are actually super duper important, actually, because of the following reason, and they're going to be important for another thing in chapter 4, but for right now, think about it like this. The microtubules actually support the movement of your cell. The microtubules can rearrange themselves, and they actually act as a highway system. So think about it like this. Um, for movement, these microtubules will end up kind of stacking one another, and as they stack to one another, they're going to be pushing the cell in a certain direction. Okay, that's how your cells actually get to move. Um, and also, they add like a highway system, okay, in your cell. What does this mean? Remember all the way up here, I was talking about how the rough ER has this vesicle that gets out, and then it goes all the way to the, um, what you call it, the Golgi Complex. Well, the way it does that is because it has to have a transport system. This transport system comes from these wonderful cytoskeletons. They're also made up of intermediate and microfilaments. On them are a bunch of motor proteins that use ATP to move around, and your vesicle ends up sitting on one of these motor proteins, and through the cytoskeleton, it'll be transported to its intended destination. It's also how once you leave the Golgi complex, the vesicle will hop on another transport vehicle or a transport protein, and that transport protein, using ATP, will end up going to the outreaches or the plasma membrane of the cell by using cytoskeleton. So think of cytoskeleton as a literal highway system and also used for movement when they stack upon one another and they kind of push the cell in a certain direction. That is a more hypothetical thing. Scientists are still experimenting on that movement portion, but it's something I guess you could maybe use, who knows, when they get added to the curriculum. Next up, central vacuole. These are mainly in plant cells. They are used basically to hold water. When you have too much water, they flush water out, and when you have too little water, they just try to store as much as possible for your the plant's benefit. If you think of a plant, and by the way, this takes up the majority of the volume of a plant. Um, it's the following. Here's my vacuole, super big like this. It's all water in here. And as it gets bigger, because my plant needs water when it rains, it'll actually exert pressure onto the cell wall, and I'll explain about that in the following section of your chapter. Anyway, centrosomes, no need to know that. It's important for mitosis. I'll skip along. Plasmodesmata, super important. You will absolutely be getting a question about this. This is how when plant cells are next to one another, they want to basically send molecules, all right? It's an efficient highway system, basically. It's an example of juxtacrine signaling. You'll learn about that again in chapter 4 or unit 4. But basically what it is, it's these small little holes, like little openings of a cell that kind of combine two cells together and allow polar molecules to go through one another. That's literally it. Okay? If I were to show you right now what they look like, Plasmodesmata, boom. Super, super small. They just allow, and they're like little crevices, they allow for things to kind of go through the plant cell. Know that, super important. Ions get through. Know which organelles are membrane bound. Membrane bound organelles are basically everything I've listed, so all of them. know which organelles have a double membrane: chloroplast, mitochondria, and actually the nucleus as well. I forgot to mention that. My apologies. Given the fact that ribosomes are found in all life forms, why is this meaningful? It basically means that we all have a common ancestor, because if we all have ribosomes that can read our DNA, probably we originated from somewhere. What is the endomembrane system? Bingo, this is super important. The endomembrane system, if you remember how I said that the rough ER, right, goes to the Golgi, right? Well, this is kind of like a system that works together, like how, it's like a factory machine, right? So the endomembrane system is super important. They're called membranes because all these functions, or all these organelles, have membranes. But if I have my nucleus first, my nucleus sends out these things called mRNA, and because it sends them out and the rough ER is right onto it, remember what the rough ER has. It has these ribosomes. The ribosomes read the mRNA and they will chug out a protein, and remember the protein will then hop onto a vesicle, which will hop onto a cytoskeleton, get transported to the cis phase, the cis phase of a Golgi. The vesicle gets absorbed into the Golgi, so like a little circle gets absorbed. Then the Golgi begins to package everything, right? Add a little bit extra stuff to the protein, and then shoot it out in a vesicle, which will then end up on the highway system, aka the cytoskeleton, on a motor protein, and go all the way to the plasma membrane, where then, once it's on the plasma membrane, let's say right here, it gets shot out and goes to its intended area. Boom. So, what is the infolding membrane bound theory? You will not be asked about this. This is like just something that the book always has. What is the endosymbiont theory? This is huge. You have to know this. The endosymbiont theory, or symbiont theory, I think it's symbiont. Yeah, symbiont theory is the following. It is the belief that our mitochondria, our mitochondria, and and that's a and our chloroplasts, well, not our, but chloroplasts, are from prokaryotic cells. And really, the belief or the story that goes along with this is, a long time ago, there used to be these things called proto-eukaryotic cells. These are before our eukaryotic cells were really made into like a lot of organelles. These proto-eukaryotic cells were these really, really huge cells. And if you remember, huge cells were inefficient because of the following. They had a very poor surface area to volume ratio, but they still needed to get energy, right? And what basically happened is that other bacteria, right, these prokaryotic bacteria, were around, and what they could do is they could make energy from sugar in the case of mitochondria, or from light in the case of chloroplast. And what happens is that a mutualistic, or maybe even a parasitic relationship occurred, where the sugar ended up actually going into, or not the sugar, sorry, the prokaryotic cell ended up getting engulfed into the larger cell. And what happened is the larger cell clearly wanted to eat the smaller one for energy, but remember, it was inefficient. But as it engulfed this prokaryotic bacteria, what ended up happening is that the prokaryotic bacteria did not die. As a matter of fact, this prokaryotic bacteria stayed in there, and because it could provide energy from sugar, it would provide energy for the large cell. In return, it would gain protection from the um, from the outside environment, right? It's inside the larger cell. Larger cell has bigger stuff, more protection devices, more proteins. It gets protected. Boom. So that's the story behind it. But where is the evidence? And this is where you're going to be really getting get tested on. Where is the evidence for this? Well, if you were to do a dive into the DNA, the DNA of the human body, or in any eukaryotic cell, the DNA of any eukaryotic cell, you will see that it does not code for mitochondria or or chloroplast. And as a matter of fact, what happens is that these chloroplasts and these mitochondria have their own DNA and they replicate by themselves, which makes sense because they were old prokaryotic cells, right? And prokaryotic cells had to divide themselves. So when it's time for mitosis, the mitochondria triggers its own division system. And its division system, ironically, is very similar to how prokaryotics divide. You don't really have to know how prokaryotics divide, but it's called fission. Mitochondria do that. In addition, they have their own DNA, and their own DNA is circular. And if you remember from a while ago, in this video, I said that circular DNA was from prokaryotic bacteria. So, with that being said, that's another thing that they have. They have circular DNA instead of linear DNA, which is similar to prokaryotic bacteria. Okay, circular DNA. Interesting. Now, the other thing that was pretty interesting, and this is somewhat of an easier one, is that they have similar size. They straight up just have similar size to a prokaryotic bacteria. Remember, prokaryotes are smaller than eukaryotes. Makes sense how the eukaryotes engulf the prokaryotic cell, aka the mitochondria. That's another thing. So, the size. And lastly, if I were to look at the layers of the mitochondria or the chloroplast. And what I mean by layer, I mean the membrane layers. Remember, they have two membranes, right? If I were to look at it, I would notice that the inner layer of the mitochondria is actually composed of similar proteins and similar structures to prokaryotic DNA. So the inner membrane, aka the cristae of a mitochondria, or the vacuolate, cristae, or the vacuoloid of a chloroplast has similar structure, aka proteins, to prokaryotes, and its outer membrane has a similar structure to eukaryotic cells. And that makes sense because eukaryotes engulf these cells and they kind of just added their own thing to the extra region. Eukaryotics. Boom. So that is your Endosymbiont Theory. So yeah, there is just this huge list here too, so don't worry about it if you didn't catch everything. Next up, this is the newer or the extra portion of um, the content. This is cell transportation, and cell transportation kind of goes back to the plasma membrane, and we're going to be analyzing how the plasma membrane really works with this type of stuff. So where to start? Let me pull up this. What are the two? Okay, yeah. Based on the structure slash properties of the cell membrane, you should know the relative permeability of small slash large and polar and nonpolar molecules. Again, small molecules typically are going to pass easier through the membrane because they can kind of go through the cracks and crevices of the hydrophilic heads. And then the larger the molecules, it's going to be a little bit harder, but remember that nonpolar molecules will always go easier through because they are the most permeable from the fact that the hydrophobic interior is the largest portion of the plasma membrane, allowing the nonpolar things to kind of really be attracted to that part and go through. Oh, here, perfect. O2 and CO2 are classified as what type of molecules? They're small, nonpolar molecules, so small, nonpolar. You just have to memorize that. I'm not going to get into the chemistry of it. The video is already too long. Don't want to bore you guys out. Small nonpolar molecules, they go through simple diffusion, and I'll explain what simple diffusion is. Anyways, I have the list here from what I described earlier. How does water primarily pass through the cell membrane? Some of the water, okay, can pass through simple diffusion, which is just like naturally going through the plasma membrane. Okay, some of it can, but most of it goes through those proteins that we're talking about that are on the plasma membrane, and those specific proteins for water are called aquaporins. Now I'm going to touch about things like diffusion. Okay, diffusion is basically things spreading out. Okay, going from order to disorder in a way. But if I were to have a box, right, and I were to put everything in this box clumped together, and these are molecules, right, clumped together right here, eventually the molecules are kind of spread out, right? Diffusion works in most cases when I have the following. I have a membrane or some barrier that is permeable for molecules, and I put a lot of molecules, in this region right here. And I'm going to kind of just scribble around, but I have a lot of these molecules right here. And diffusion happens when these molecules end up going towards the other end that is empty much quicker at first, because really they kind of begin to push one another around and shove them out. But they want to always try to get, well, molecules always want to be in this thing called equilibrium, all right, where diffusion is really what is happening because of this, right? Molecules want to go through equilibrium. Molecules want to be in equilibrium. Not too important for biology right now, but they want to be in equilibrium. And what they do is they try to make the rate equal to one another. So what I mean by that is when these molecules begin diffusing through, right, they're going to be diffusing at a very, very fast rate. But as the concentration decreases, right, there is the molecules that are here, right, are going to bounce back into here, and then the molecules in here are going to bounce back into here. And when they reach that same rate, that is what diffusion is trying to get. That the rate of molecules going through that permeable layer, or that permeable barrier, is equivalent to the rate of the molecules coming back into the original spot. All right, but diffusion overall is just things passing through, but its intended goal is to reach that equilibrium, that equal rate of, let's say I have, really visualize this permeable barrier right here. I have like 4 molecules. When the speed at which these molecules, right, it's going to be very quick at first because I have a lot of molecules here and nothing here. When all these molecules try to go through to this extra end, and the rate at which the molecules that have ended up in the extra end kind of come back, right? That when that rate is equal, you've reached equilibrium. That is what diffusion is trying to do, right? Trying to reach that equilibrium, but overall it's just the spread of molecules to the other end. Okay? So when I now say things or definitions like simple diffusion, okay? Simple diffusion means molecules going, and I'll actually begin to type this out. Molecules that go through the plasma membrane with ease. So these are always nonpolar small molecules, all right? They go through with ease. They simply just go through it, okay? Then you have um, facilitated diffusion. Facilitated diffusion can be broken up into two things. And now, facilitated diffusion means that you are helping things diffuse through the plasma membrane because they originally could not. I spelled could incorrectly. Could not. Okay? What does that mean? Basically, it means that I'm using a protein to get things through. Now, there are 2 types of facilitated diffusion. One is called passive transport, and the other is active transport. And there are 2 main proteins that are associated with these. Passive transport has these things called channel proteins. An aquaporin is a channel protein. So if I were to draw my plasma membrane, right, and I have these phospholipids here first, and I were to draw my protein, it is going to look like a channel. There is literally going to be an opening and a closing, and things just kind of just go in and go out. They just shoot. It's a channel, like a tube, right? And this requires no ATP because things will just naturally go down or naturally go up, okay? I'll describe when they want to go up and down, by the way. But when the rates are equal, they'll just kind of be going at the same rate because they've reached equilibrium. Active transport, and I really butchered this, the writing of active transport, requires ATP. Okay? So there is a caveat here. For passive and active transport, there's a very easy way to distinguish between them. Besides their proteins, it is the concentration. Concentration. You have to identify concentration. Remember how I said that the rates in an equilibrium are equal, and how at first one rate is super higher because there's a lot more molecules in one area and there's nothing in the bottom area, and they try to go to the smaller area? Basically, like if you looked at here, there was no molecule here, so the rate of the molecules going into this empty area is way higher than the rates of the empty area going back in because there's nothing there. I know that sounds redundant, but hear me out. Basically, what diffusion is trying to do is going from a high concentration to a low concentration, right? And trying to equalize both areas. That's what I mean by reaching the same rate. The concentrations are going to be equal, kind of. Um, I'm not going to get into the chemistry of it again, but just think about it. Diffusion always wants to go from high concentration to low concentration. It also makes sense or biologically speaking because, look, you have all these molecules kind of combining one another. They're going to collide and push themselves to the area of lower concentration where there is really no molecule. And if I were to draw my barrier, that kind of makes sense, right? These molecules hit one another, they push, they kind of go into here. It's making a very long video. I'm so sorry, guys, but this is almost towards the end. But in passive transport, you're typically going to get things that are going from from high to low concentration. High to low concentration. It's natural, and you're also going to be using it you're going to be really using it for mainly polar molecules. The same thing is, by the way, true for active transport. You will be using it for polar molecules as well. Okay? And the main thing here is that it is unnatural. Remember how things like to go from high to low, like that I just said? Well, active transport is when you go from low to high, right? You're actually going to need a thing called a barrier or a gated protein. And basically, these proteins will only get activated if they have ATP attached to it, and they allow the things at a lower level or at lower concentrations go to high concentrations. So active transport, low to high concentration, passive transport, high to low concentration. The reason why, high to low is a natural thing. It's just easy. You don't really molecules want to do that. Low to high concentration, you need energy because this is unnatural. When things are unnatural, you kind of have to go against the force of nature. That requires energy. So let's say if I have 3 molecules here and I have 5 molecules here, and I want to push these 3 molecules through, I'm going to need ATP, and this is the most important part of active transport. I'm going to need ATP to turn on this barrier protein, okay, activate it, so it kind of changes its shape a little bit. Remember, these channel proteins on the passive transport, they're just open. These barrier or these gated proteins are not open, okay? They need ATP to kind of turn on, and then they open up, right? They'll open up temporarily, like let's say like this, and I will draw the opening here, right? So let's say the ATP hits it, right? It transforms it. Some of the molecules, right? 2 of them can go through. Now you have, 7 molecules on top, assuming that you had, 5 here, right? And you have maybe 1 down here. Okay? That is the active transport, that is the passive transport, and that is simple diffusion. Okay? So I hope that makes sense. It's still all here. There is a thing called osmosis. Osmosis is the simple diffusion of water. Okay? Just simply related to water. Simple diffusion of water. Okay. And really, what you're going to see osmosis used in is the following. You're going to be used, or you're going to be, well, actually, let me describe it easier. There is this classic model, and you're going to be using it in a thing called identifying tonicity, or osmoregularity. Okay, so you're going to be using osmosis, the diffusion of water, right, to identify these things called tonicity or osmoregularity. Big important part of the test, by the way. So you should really get osmosis down. Again, diffusion of water. Basically what happens is the following. If I were to have water that is very, very, like let's say I have water and solute. Solute is just something that binds to water. Let's assume that these black things are all water and I have very minimal amount of solute. What I would call this type of solution, where I have high water, H2O concentration, H2O concentration, and low solute concentration, things that attach to water. By the way, this is like in your normal drinking water too. Low solute concentration, I call these hypotonic. Hypotonic. And the reason I would identify this as hypotonic is because, well, there's a lot of water here, very low solute concentration here, so then it's going to have its opposite as well, right? I'm going to be drawing the water here, and in the other section of this pool of water, there is going to be a lot of solute. A lot of solute. And what this means is that this is going to be called hypertonic. A hypertonic solution. And it is going to be characterized as very low H2O not very low, but low H2O concentration with high solute concentration. With high solute concentration. Okay. Right now, on purposes, I'm going to be showing you like this in an experimental tubing thing, but if you have a lot of water and not a lot of solute, right, that thing is hypotonic, the water is more likely to go towards the hypertonic area. Why? Because there are a lot less molecules in that area, right? Because they're being consumed by the solute or being attached to the solute. Therefore, there is a high concentration of water here, high concentration of water in hypotonic, and low here. So, remember how I said diffusion always wants to go from high to low? Well, this is going to simply diffuse into the hypertonic area. The hypotonic will push water into the hypertonic region until this is the big part, until they are equal in concentration. And that is what you would call isotonic. That is a very favorable state where the rate of water going into the other area and the rate of water going into the backwards area, so like forward and backwards, is equal. Okay? So that's that. And it's because the concentrations are the same. That's osmosis. Where do we use osmosis, right? Why is this important? Is with cells. Remember, your cells are suspended in or basically in blood and other things, but there's a lot of water outside your body, right? Your body's like 99% water, 9% water. I don't know the percentage, but you have a lot of water, and you have a lot of solute in your body too. Solute, by the way, can literally just be NaCl, salt, K plus, Cl negative, Na plus, all these molecules, right? These are called solute because they can attach to water. Now, if I were to have a cell that is very hypertonic, right, that means that I do not have a lot of water into my cell, and the exterior is hypotonic. So the environment is hypotonic, right? There's a lot more water concentration in there. What's going to end up happening is that all the water is going to rush into the cell all of a sudden. And as a result of that, the cell is going to end up exploding. It actually explodes. It's called a lysed cell. A lysed cell, and these are, by the way, for human cells or animal cells. In animal cells, if you have a lysed cell, it means the environment was hypotonic, with an internal that is hypertonic. Because water keeps on rushing and rushing into the cell, and it just blows it up. And because it's at such a fast rate, by the way, it doesn't have time to reach a favorable state. If the water or if the concentration of water in the inside and the outside are the same, it's just called isotonic, and you have a very favorable condition for the cell. You want to be isotonic. Your cells are in an isotonic state. And next up, if you have a water, or if the concentration of water is in a hypotonic area, meaning that there's way more water in your cell than solute and there is more solute outside your cell than there is water, the water will begin to rush out of your cell, making it shrivel up because water holds up space, right? Everything in your body holds up some space if there's something inside of it. So the cell will end up shriveling and dying. So you will have a shriveled cell if you have a very hypertonic environment. Now, you're going to see this on the test probably, where you are given a patient who needs IV immediately, and you want to give them IV so that they can reach a isotonic state, or you want to, you want to basically make sure they're isotonic. You would have to basically evaluate the environment of the inside of the human, right? So see like the amount of solute they have, and say like, okay, if there's a lot of solute in the body, I probably want to, go with maybe a more moderate hypotonic solution so my cells don't blow up, right? I that's basically it. Um, so yeah, there's that, and these actually end up doing different things in plant cells. In plant cells, if you have a hypertonic environment, right? If you have a hypertonic environment, what's going to end up happening is your cell gets plasmolized, right? Because you assume that the interior of the cell is very hypotonic. And if it's hypotonic, meaning there's a lot of water, the water is going to leave. And what ends up happening is that the cell wall actually gets detached from the membrane. Because there's like these gluey things connecting the membrane and the cell wall. And when all the water is leaving your cell, you're actually gonna be like shrinking, right? And when you shrink, you actually rip off, like you shrink so fast that you rip off the cell wall from the cell membrane. So that's called plasmalized. Then when you have an isotonic structure, you actually are in a kind of in a what's the word? Isotonic plant cell. You're kind of like in this. It's called flaccid. That's the word, flaccid. Where if you can imagine plants that haven't been watered in a day, they don't have that much water in them, but they're still kind of isotonic. The plant's gonna kind of be like dead a little bit. It's not gonna be dead, but it's gonna kind of like be flat because what happens is, remember that all the water goes into the vacuole of the cell. And basically what happens is if there's like not a lot of water, but a decent amount that some pressure is getting exerted onto the cell wall, the cell is going to kind of just be in like this like thinner state, right? As you can see here. It's going to be like, you see like these crevices right here, right? That's flaccid. But if I fill my water up, or if I fill my cell up with a lot of water, meaning that the environment is hypotonic, so hypotonic environment, I'm going to have a type of plant cell that is turgid. Turgid. What this basically means is that as I have all my water flowing into my cell, my vacuole, my central vacuole gets so huge as it expands that it's going to be placing pressure onto my cell wall. Now, will my cell wall burst? Possibly, eventually it could if you put a lot. But there's like this counter pressure that actually ends up happening from the cell wall that it prevents the cell from kind of blowing up and becoming lysed. So you're going to have a turgid cell. So a turgid plant, what's that look like? Like this. No, not like that. Like this. Flaccid plants are like that. Plasmolized plants are dead. Okay, so that's basically that. So moving on. Moving on. I do believe that we are very close to the end. I have described facilitated diffusion. I have described how diffusion works, simple diffusion, facilitated diffusion, depending on what you need. Active transport I have described, going from low to high concentration. What are the different types of active transport? This is the last part of your lecture today. Types of active transport. So basically any type of thing that requires ATP. You're going to have the following. Exocytosis, meaning exiting the cell. Exiting the cell. And by the way, this is the end goal of the endomembrane system, right? To get stuff out. Or you have endocytosis, which means entering the cell. End, enter, exo, exit, enter the cell. Basically how this works is that once you've gotten your molecule, the vesicle that's, the protein that's in the vesicle on these proteins that move around, they're called transport proteins or motor proteins. They kind of move around. They bring your cell, by using energy, all the way to the plasma membrane. What they do is that vesicle, which is made of by a phospholipid bilayer, it's going to be absorbed into this phospholipid bilayer that's part of the plasma membrane, and proteins are going to be basically used to kind of put everything, rearrange stuff, and push that protein actually out into the environment of the body. Endocytosis is basically the same process. As a protein comes and enters a cell, it's going to go and it's going to create its own vesicle around it to be mediated in the cytosis, and it's going to have ATP used. Next up, you have phagocytosis, pinkocytosis, and receptor-mediated endocytosis. Phagocytosis is basically when a cell absorbs something. Pinkocytosis is when the cell absorbs a liquid, and it's basically just endocytosis of something, but that it's just more specific endocytosis, and it's going to be a more specific type of exocytosis too. I don't think your study guide talks about this, but I think that it's very important to know. It's called cilia. And I wanted to kind of talk about this before I end the lecture. Cilia are these portions of your cell that are like these tails, okay, that move around. You don't really have to know the structure of the cilia, but know that it's used for really two things: moving molecules up and down a cell wall, or a cell or a tissue, really moving molecules around. And what I mean by that is that as these things wag their tails, right, molecules are going to be like sitting here and they're going to be moved up because of that, or moved down. And the best way I can explain this is in your lungs, right? In your lungs, um, God, I'm awful at drawing, but in your lungs, you're gonna have these lung cells, and they're gonna have these cilia that kind of push mucus all the way up, right? All the way up to where you can cough it out, right? That mucus is those molecules, and it travels along the cilia to kind of get pushed out. Also, if you think about cilia, they have these like longer cilia, these one type of cilia, that is used by sperm to move around. Cilia is also used for movement of a actual cell. Typically sperm cells, by the way. And sperm cells use a lot of energy, so if you can identify that it has cilia and it is using a lot of energy, it's probably going to end up being a sperm cell. Again, remember, always try to identify, if I can make you take away one thing for this chapter, always try to identify, what the cell is really doing, or what concentration of organelles it has. You can kind of identify the purpose of the cell. So if it has a cell that uses up a lot of energy, you know that it's really either used for digestion, breaking stuff down, or it's going to be used as a sperm, which is, it uses a lot of energy to move around. So stuff like that. Um, that's that. What else do you need to know? Here's a quick thing about active transport, passive transport. So yeah, there's that. Water potential. Literally, just please, for the love of God, um, use your uh, study guide, and your notes or your equation sheet. It is literally going to be on every single test you have, the equation sheet. So you should not worry. Always look at it if you get something about water potential. You literally do not need to know this. I do not know why it's there on AP Bio. You they literally have almost no questions on it, but you may get a teacher who ends up putting maybe an FRQ about it because I know some of them do exist out there. Be careful, but that is that. Um, However, this is basically the end of the chapter, or the unit. So if you have any other questions, feel free to message me, um, add a comment on the YouTube video. But I hope this video helped. I'm so sorry for it being long. Good luck on your AP Bio test.